Gluten analysis. Test procedure for the competitive ELISA RIDASCREEN Gliadin competitive. Hydrolyzed and fermented food samples contain small gluten fragments which are detected only with the competitive ELISA. RIDASCREEN Gliadin competitive has been validated in an international collaborative study and is now official AACCI method. The kit contains all reagents to carry out the test procedure. Unpack all reagents from the box and bring them to room temperature before use. This takes approximately 60 minutes. Insert sufficient number of strips into the microwell holder. Leave unused strips in the aluminum bag so that the strips are not exposed. Now we continue with the sample dilution. First, dilute the Gliadin diluent. Add 1 ml of the concentrate to 4 ml water. The sample was extracted with ethanol as described in the test kit insert. Now the supernatant of the extract is diluted 1 to 50. Always make sure the volume settings of the pipette are correct. Pipette 20 microliter of supernatant to 980 microliter diluted Gliadin diluent. Always flush the pipette before use. At the end, mix. Use the diluted sample extracts immediately in the assay. Mix the standards and samples before use. The conjugate is diluted 1 to 11. Add 200 microliter concentrate to 2 ml distilled water and mix gently manually. Pipette 50 microliter sample or standard per well in duplicate. To save time, all vials are already open. Then pipette 50 microliter diluted conjugate using a stepper. Discard the first aliquot before pipetting the ELISA. Carefully rock the plate to mix sample and conjugate. Incubate for 30 minutes at room temperature. Put a lid on the plate. After the incubation, the plate is washed 3 times. The wash buffer has been diluted 1 to 10 in a clean bottle to which an 8-channel manifold dispenser is attached. Make sure all bubbles have been removed. Firmly press the frame to ensure strips will not fall out during discard. Discard the liquid from the wells using an angular and forceful motion to properly remove all liquid content. Vigorously tap the inverted plate on a dry and absorbing surface to ensure that all liquid content has been removed from the wells. Fill the wells evenly across the plate with 250 microliter wash buffer. Discard the liquid from the wells using an angular and forceful motion to properly remove all liquid content. Repeat the washing step 2 more times. Also make sure to absorb any liquid from the plate frame and outside bottom of the wells. At the end, visually inspect the wells for residual wash buffer or bubbles and repeat tapping if necessary. After washing, add 100 microliter red chromogen pro to each well with a multi-stepper. Avoid splashing. Incubate 10 minutes in the dark at room temperature. After the incubation, add 100 microliter of stop solution to each well with a multi-stepper. Avoid splashing. Measure the absorbance at 450 nanometers within 10 minutes. Use the RIDASCREEN Win for calculation of results. Final message. Avoid any airborne contamination from cereals. Keep windows closed. Control with RIDA Quick for gluten contamination. During assay procedure, have a continuous workflow. Avoid splashing while pipetting carefully. Washing steps are crucial and have to be carried out thoroughly. Run check samples, for example, samples spiked with VG-PAT material, with known concentrations for quality control. For beer extraction, use fish gelatin and PVP. Now you have learned the handling of the ELISA for food analysis. In the next steps, you will have to validate unknown samples in spiking experiments. Thank you for your time.